Now we have to study about the basic purpose of the cell membrane, how it functions, basic function. So this shows that the middle, this is the core, this is the core of the cell membrane, or middle side of the cell membrane. This is the core of the cell membrane, and you can see that in the core of the cell membrane what will present, the hydrophobic fatty acid stays are present. So in the core what is present, the hydrophobic means water repellent, which are non-polar, water insoluble, hydrophobic and fatty acid tails are present. So the substances which are water insoluble, or small substances can cross the plasma membrane or cell membrane easily, while the substances which are water soluble or giant substances, or ions like sodium, calcium, chloride cannot pass this cell membrane easily, because these are hydrophobic. They will repel the hydrophobic will attract this hydrophobic, while these hydrophobic will repel the hydrophilic substance, which will come and gain entry into the cell membrane. For example, if sodium ion will know that sodium ion concentration in the ECF, extracellular fluid, is approximately 140 millimole per liter, while sodium ion concentration in the inside of the cell are only 10 millimole per liter. So we will need definitely the sodium ion into the inside of the cell. So the sodium ion have to pass into the inside of the cell to maintain the cell at the normal level. So what will happen, the sodium have to move inside the cell, or when sodium will increase inside the cell from a level 10 to for example 20, then the excess sodium 10 millimole sodium have to be sent outside the cell, So how will it happen if there is also the cell membrane present? Cell membrane is present at the whole side. We know that the cell membrane is repelling. Cell membrane, according to this, the cell membrane can't allow two type of substances, one which are very large, for example insulin have to become in the cell to decrease the level of the glucose, otherwise the patient will be hyperglycemic, the diabetes mellitus will induce. So insulin has a very important function. So the large substances and the charged substances, according to this structure, cannot pass through the cell membrane, while those substances which are small, and uncharged means non-polar, can pass it easily. And I have discussed the reason, because the core contain the hydrophobic fatty acid, mean non-polar fatty acid. So the non-polar substances means uncharged substances which are small can pass this membrane easily and gain entry into the cell, while the substances which are giant, as this substance cannot enter into the cell, because this substance is giant, this substance is large, and also the polar substances like the sodium, like the calcium, like the chloride ion can't enter into the cell, because the sodium have the positive, calcium has the positive 2, chloride has a negative 1 charge. So because of the charge, because of the polar nature, it can't enter into the cell. So there will be the specific mechanism, there will be the specific pore, there will be the specific structure through which these substances have to enter into the cell or outside the cell, otherwise the whole metabolism of the body will be disturbed. So there will be a mechanism, and there exists a mechanism, we will study it later. So now we basically studied that the large and uncharged substances can't cross the cell membrane easily, while the small and uncharged substances cross it easily. So on this basis we can call this cell membrane as selectively permeable membrane or differentially permeable membrane. What it means? Selectively permeable membrane means it is permeable, it will allow substances, only selective substances, means only selective substance will allow to enter into the cell. And with selective substance, the small molecules like oxygen, carbon dioxide can enter into the you know that the carbon dioxide dipole moment is 0, carbon dioxide north polar. So the carbon dioxide, sodium, which are the small molecule, can enter into the cell easily and gain out of the cell. We know that the oxygen gain entry into the cell and carbon dioxide is expelled out of the cell to maintain the regular metabolism of the cell. So it is also called the selectively or differentially permeable membrane on the basis of its function, on the basis of its allowance to the respective ions, respective substances. So how will these substances gain entry into the cell or outside the cell, like the charge and large substances? What is will be the mechanism? So first we have to discuss about one more thing about the structure of the cell membrane. These are the phospholipid heads and these are also the phospholipid heads, and beneath these phospholipid heads ICF is present, mean intracellular fluid, means these lies inside the cell, and these lie outside the cell because ECF is present. And head is always accompanied by its tails, which may be unsaturated and saturated. I draw this as this may be unsaturated, saturated, unsaturated, saturated, unsaturated, saturated, 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 unsaturated. So these are the hydrophobic end and these are the hydrophilic tail or polar tails. So this is the outer leaflet of the cell membrane, while this is the inner leaflet of the cell membrane. We can call this as outer leaflet of cell membrane because it faces the ECF, and we can also call at the same time this as the inner leaflet of the cell membrane because it faces the ICF, interstitial fluid, intracellular fluid. So what will happen? The carbohydrates abundantly present in the outer leaflet, whereas there is no carbohydrate or very minimum carbohydrate are present in the inner leaflet of the cell membrane. So the first difference between the outer leaflet of the cell membrane and the inner leaflet of the cell membrane is the abundance of the carbohydrate, presence or absence of the carbohydrate. So at the outer leaflet, carbohydrates are present abundantly, carbohydrates are present, while in the inner leaflet there is no any carbohydrate, very minimum, no any carbohydrate. So this is the basic difference of carbohydrate. And there is also a difference of the proteins. We know that these are the phospholipid and the proteins will attach to these lipids in a mosaic manner, means this will be the protein, this will be a protein, this will be a protein, this will be a protein. We'll discuss that. This is the integral protein, which is also known as the transmembrane protein. These are the peripheral protein. We'll discuss it later. So the proteins which are present in the outer leaflet and inner leaflets are also different. So the proteins of outer leaflet and inner leaflets will be different. So first we discuss about the difference of the outer leaflet and inner leaflet. About the abundance of the carbohydrate, the carbohydrate are very abundantly present in the outer leaflet, whereas the no carbohydrate are present in the inner leaflet. Then we discussed about the proteins present in the outer leaflet of the cell membrane which faces the extracellular fluid are different from the proteins which are present in the inner leaflet which faces the inside of the cell or the ICF. Then the third major difference between the outer leaflet and the inner leaflet is the presence or the abundance of phospholipids. So in the outer leaflet, two types of phospholipids are mainly present. In the outer leaflet, two types of phospholipids are mainly present. One is the phosphatidylcholine. These are not much difficult name because these are named as I discussed that phosphatidyl means this is the phospholipid or phosphatidic acid in which a choline nitrogenous base, which is a nitrogenous base, is attached at the phosphate side. And then the second most abundant phospholipid in the outer leaflet of the cell membrane is sphingolipids. The second lipid which is present abundantly in the outer leaflet is the sphingolipid. And what type of the lipids are abundant in the inner leaflet? So inner leaflets contain phosphatidyl Then the second is phosphatidylserine. These are the abundantly. So the first difference is the carbohydrate are present abundantly in the outer leaflet of the cell membrane, whereas no carbohydrate is present inside of the leaflet. While the second difference is the proteins. The protein present in the outer leaflet of the cell membrane are different from the proteins present in the inner leaflet of the cell membrane. And the third major difference is the abundance of the lipid. The most abundant lipids which are present in the outer leaflet of the cell membrane are the phosphatidylcholine and sphingolipid, while the most abundant lipids which are present in the inside of the cell membrane are phosphatidyl ethanolamine, phosphatidyl serine, etc. So the next, as I told you, that the large and charged molecule can't pass these membrane easily because the core is the hydrophobic fatty acid, which will not allow the charged or large molecule to pass through it easily. So there will be some other mechanism that will allow. There are some proteins. Assume this is a protein. There are some proteins in the cell membrane through which these large, for example, insulin or large substance or ions, for example, sodium, can pass through those proteins molecules and enter into the cell. So how the large and charged molecule gain entry into the cell? There are at a certain point of the phospholipid bilayer there will be the protein present. We'll discuss it later that these proteins are the transmembrane protein, which are also known as the integral protein. Through these proteins the substances enter into the cell and reach the interstitial fluid. And the same is for the opposite mechanism. The substances which are formed in the ICF, which are larger and charged, can't pass through outside, will pass through these protein channel and will go outside the cell. We discussed that these proteins may be channels, may be carriers, may act as enzymes, may act as a linker protein, may act as receptors. These are all about the types of the integral or transmembrane protein.